BioBox provides you with a collaborative platform to organize, store, process, visualize, as well as explore key biological insights from your RNA sequencing data from start to finish. Today, I'm going to walk you through a step-by-step tutorial of how to go from raw FASTQ files to process differential expression as well as gene count files with visualizations such as heat maps, volcano plots, and most importantly, key biological insights from your data. To get started, the first thing that you have to do is set up your library. Your library is where you'll store all of your files, as well as the information associated with the samples used to generate those files. The library can be accessed from the top left-hand menu. And the first thing that you have to do is configure or create a model. On the BioBox platform, we do have two example models, patients as well as cell lines. Models are the overarching category that connect all of your data sets, and they refer to the model organism that was used to generate the data. So for example, human cell lines or human patients, or mouse cell lines, for example. And all of the information associated with each model can be completely customized to your liking. For example, if you are tracking a specific mutational status or a treatment that was added to a cell line, all of that can be customized within the BioBox library. So the first step is to either configure one of these models or create a new one if cell line and patient don't suit your needs. For this example, we'd be working with some breast cancer cell lines that we would like to do RNA sequencing on. So the first thing that I would like to do is to configure some of these model properties to ensure that they adhere to all the properties I would like to keep track of. And to do that, I would go to the Configure menu and edit Model Properties. Here I have the option of removing or adding dictionary terms, which will be the properties that are tracked for each cell line that I have. If I wanted to track a specific mutation or any other category that's not found in the dictionary, I can create a new dictionary term, and it can be a free-form text, a numerical field, a date, a link property, for example, linking a cell line to a patient that it's derived from, a Boolean, which would be a true or false object, as well as a drop-down. For this example, I'll go with a drop-down. And let's say I wanted to keep track of a specific mutation that was in several of my cell lines. And here I would add all of the fields that would be associated with this drop-down property. And if I would like to, I could also allow for multiple values to be selected, so perhaps the cell line has more than one mutation. Once I'm satisfied with the fields, I can hit Submit, and the new dictionary term will populate here, and I can simply drag it into the Custom Properties section and it'll save. If I don't want to keep track of, or I'm not interested in any of these fields, I can remove them by clicking the X. Once I'm satisfied with my model, I can save it, and now I can see that the model has been updated with all of my new properties. So now I would add in the cell line entities, and these would be all of the cell lines that were used to generate the RNA sequencing data that I'm working with. To add more than one cell line at once, I can go to the Bulk Import button, and here I'll be taken to a table where I can just copy and paste all of these values in from Excel. Once I've copied and pasted all of my values in from Excel, I can hit Submit, and all of my cell lines will be generated. So now I can see every single cell line, as well as the information such as the mutational status or the treatment that was applied to these cell lines. The next step is to create your samples. So your samples are your analytes, either the DNA or the RNA that was used to generate these sequencing files. So if we are working with RNA sequencing, we would go to the RNA tab, and again, go to bulk import so you can add more than one sample at once. And here you would just specify what the sample would be called if you had unique identifiers either sent to the sequencing facility, or these can be identical to your model entity names. Again, you would just copy and paste these values from Excel. So in my example, I was using cell lines 001 to 0010. I can copy and paste those in. The next most important step is you have to specify the analyte. So whether this is DNA or RNA, and then you can just drag this property down. And the next most important step is to specify the source. So which model entity or which cell line did they actually come from? So again, you can copy and paste the same entity names that you used in the model section to the source column. And now when you hit submit, all of your analytes will populate under the RNA tab. And if you hover over each source, you'll see all of the information associated with each cell line. The next step is to upload your data. So on BioBox, all of our processed as well as our raw sequencing data is found in the library section under the data tab. So all of the organization is done for you, and it's linked to all of your samples and all of your models. From the sequencing facility, you will receive FASTQ files, 2 FASTQ files per sample because they are paired reads. To upload a FASTQ file, simply use the Upload FASTQ File button, select the file from the computer, as well as the sample that it's associated with. So here my first file is uploading. I can close the upload drawer and I can continue to upload more files, and I can upload each file individually and just associate it with each sample, and then I'll be ready to design a pipeline and process my data. Once you've uploaded all of your FASTQ files, you can get to the fun part, which is processing your data. To do that, you can create a project in our workspace. Our workspace is located in the top left-hand corner menu, and this is where you'll have all of your projects. So this includes where you'd process data, where you're going to visualize data, as well as explore those key biological insights. So the first step is to create a new project, and you can give this any name that you would like. Once you've created your project, you'll be taken to the overview board. The cube section is where you'll actually build your pipeline and process your data. To create a new cube, go to Create and give this cube any title that you would like. Because we are working with RNA sequencing data, I can title this RNAseq pipeline. And the next step is to go to the Design tab. Here I'll have the option of creating the new pipeline that will be used to process my data. We do have some BioBox standard pipelines that you can get a preview of, so we have a star into FastQC as well as a mutation calling pipeline. But if you are processing RNA sequencing data, you would have to create a new pipeline, and from here you'll be taken to the Pipeline Design Studio. So here you'll see all of the inputs that are available. So you can build a pipeline with FASTQ inputs, with BAM inputs, as well as sample gene counts. For transcriptomics, we have STAR, as well as DSeq2. First step in building your pipeline is dragging in or selecting which input files you would like the pipeline to run with. Because I do have FASTQ files, I'll select those, and because these are paired reads, 2 of these FASTQ files will be corresponding to 1 sample. The next step is to select the workflow that I would like these input files to go into, and that would be STAR, because the first step in the RNA sequencing alignment is to align the reads to a reference genome. To connect the workflow inputs, simply drag the red dot into STAR, and that'll connect the inputs into the workflow. Because I do have 4 samples, 2 normals or 2 references, as well as 2 treated samples, I will be doing this 3 more times. So again, these would be the paired reads for the second sample. I'm selecting STAR because that is the alignment tool, and I'm dragging in the input files into the STAR pipeline. So these would be an example of my two reference samples or my two normal samples. To make this a little bit more clear, I can also update the name. So I've updated the names, and I will do the same thing for my experimental or my treatment samples. So once I've added in all of my FASTQ inputs, as well as labeled them so that I know which ones are my paired reads, as well as which ones are my normal versus my treated, or whatever else I might be tracking, the next step is to add in the differential expression workflow. So this is going to determine which genes are differentially expressed between the two groups of gene counts, one being the reference and the other being the experimental. So for the entire RNA sequencing experiment, you only need one differential expression workflow. And from here you'll be connecting the sample gene counts, which are an output from the STAR workflow, into the DSeq2 workflow. Here you're going to select whether this is the experimental or the reference group. So here, because I can see that this is one of my treated samples, this will be my experimental group. And I will do the same for the other sample gene count files. So again, this is a sample gene counts coming from one of my treated samples. And here I have my normal samples or my reference. I'll be dragging in the sample gene counts and confirming that this is part of the reference group. So coming out of DSeq, you'll have your gene counts matrix as well as your differential expression file. And if you do want to, you can always click on the workflow to use some of these advanced configurations, as well as select the p-value adjustment method for DSeq. Once you are satisfied with your pipeline, you don't have to use the advanced configurations. They would just run according to standard protocols. And all of this information in regards to what workflows were used, as well as which files and which samples were used, will all be saved for you in a method section at the end of the workflow. So the next step is to give your pipeline a title. So this is an RNA sequencing pipeline. And now once you save this pipeline, this can be reused by yourself or anyone else in your lab should you choose to share it. In the description, you might want to specify how many samples are in the specific workflow. So for example, 2 normal, 2 treated. And then you can save this pipeline and you can exit. So now you can see that the pipeline that you just created will appear here in your drafts. And if you select details, you can see the entire pipeline that you just built, as well as all of the input files, as well as all of the output files of this pipeline specifically. If you are satisfied with your pipeline, you can launch it. And from here, you'll just have to drag in exactly which files you would like the pipeline to run with. And these are going to be files that you have uploaded to the BioBox platform. So here you can go to the arrows to expand all of your folders, and this will just show you all of the data that you have uploaded to the BioBox platform. So these are the FASTQ files that I previously uploaded, and now I'm just going to drag them and drop them into the appropriate sections. So these titles here are the titles that we gave to the input files. Once you've dragged all of your files into the appropriate sections, you can hit submit, and automatically your pipeline will begin running in the cloud. On the progress page, you can see each of the steps in real time, and as they progress, you'll also be able to see their progress here. And the same goes for the DSeq workflow. So as you can see, STAR just began to run, and if there are any issues or any errors, they will appear at the top here. Once your pipeline has fully completed, you'll be able to access the results on the results section. And now if you go to the details page, you get a full description of exactly which tools or which workflows were used to process your data. If you did use any advanced configurations, all of that information will be found here, as well as exactly which samples and which models were used to generate this data. So now we just have to give the pipeline some time to run, and once the pipeline is finished, we can get into the fun part, which is visualizing the data as well as exploring the biological insights. Once your pipeline is complete, you'll be able to head over to the results section. On the results section, you can click through each workflow, and you can look at all of the individual steps as well as the output files. All of the files generated from every single workflow within your pipeline are saved directly to the BioBox library, so you don't have to worry about exporting or downloading any of the files. You can save them and work with them directly on the platform. Open a BioBox to visualize your data using a heat map or a Volcano plot, and you can also look at your insights. To do that, head back to the main project page. Select BioBoxes and create a new plot. You can give this plot any name that you would like. And from here, you can select the visualization that you would like to work with. I'm going to start with a heat map. From here, you can select the file that was generated from either uploading a process dataset or running a pipeline on our platform. And you can see the samples that were used to generate this file. And you'll select a gene counts file for your heat map. From here, you'll be taken to a data table which contains the gene count matrix. Click on Heat Map to begin working with the data. Once the heat map loads, you can fully customize all of the properties associated with your heat map. What you might want to do first is select or add on any metadata. For example, if you were running an experiment and you were looking at cells that were either treated with a specific drug or cell lines that had a mutation, if you've added this information into your BioBox library, all of this can be displayed on top of your heat map. Simply select Add and select the type of metadata that you would like to add. Here I've added on the mutational status of my cell lines. If the cells were also treated with something, I could also add that specific treatment on, and it's accompanied by a legend. At any point in time, I could change the colors of the metadata associated with my legend. And I can also remove them at any time by clicking the X. Next, I can adjust the cluster depth to make the clustering more or less stringent. You have the option of sorting by variance or by sum, and you can also reduce the number of genes that are displayed in your heat map. If you do have 100 genes, when you export them or download the plot, all of the gene IDs will be resolved. So let's say we wanted to take a closer look at our genes and we wanted the top 50 sorted by variance. So now my heat map has been reduced to 50 genes. If I would like more information about any gene, I can hover over and I can get a description, if it's available, about what this gene does. Additionally, I can hover over each tile and I would get the Z-score, as well as the gene that it's associated with, as well as the sample type. You can also customize the colors of your heat map, and you can change it to any color that would best suit your needs. Once you've finished customizing your plot, you can take a deeper look at the genes that are involved in your heat map by looking at the insights. We break down the insights on a clade by clade basis, so that means you would get a gene set enrichment for each clade. To access this information, double-click on the clade, and you're going to get a gene set enrichment for those genes that are found only in that clade within your heat map. We break down the gene set enrichment according to various categories, including epigenetics, ontology, so this includes things like biological processes, molecular function, as well as cellular component from gene ontology, diseases, which includes OMIM diseases, as well as MSIG-DB, disease, which includes things like OMIM disease, as well as MSIG-DB. Tissues and cells includes information from the Tissue Protein Atlas, as well as GTEx. Domains include gene set enrichments from PFAM as well as Interpro. And pathways includes reactomes. So for all of these different gene set enrichments, you can click on a specific pathway, which is represented by one of these bars, and you can get an in-depth look at which genes are enriched in this pathway specifically, as well as in the clade which you are looking at. Clicking on each gene, you can get more information, as well as any observations in all of the data sets that you have on the BioBox platform. If it's available, we do break down the pathway events, as well as provide a diagram so you can take a closer look at exactly what is going on in the specific pathway. So this is an overview of the heat map. The next plot that you would most likely want to make when you are working with RNA sequencing data is a scatter plot or a volcano plot. To do that, go back to the project main page and create a new BioBox. From here, I'll select scatter as my visualization, and now I'm going to select a differential expression file to be visualized in the plot. So you can pay attention to the file type, as well as whatever file name you have given this specific file. From here, you'll be taken to the data table, which will give you a look at the gene IDs, log2 fold change, as well as p-adjusted values. You may have additional columns, but these are the three most important values for the purposes of creating a scatter plot. So if you click on scatter, you'll be taken to the graph. The first thing you have to do is specify what is going on your x as well as your y-axis. On the x-axis, most commonly you can use the log2 fold change, and on the y-axis you would use the p-value or the adjusted p-value. And here, all of the data points in your file will be plotted. Adding tooltips will allow you to take a closer look at each gene point. For example, if I've added gene ID, log2 fold change, as well as the adjusted p-value, all of these descriptors will appear over each data point. Under axes, you can fully customize the graph. You can fully customize the x as well as the y-axis. For example, you may want to add a logarithmic scale and adjust the log base, or you could keep it as a numerical value. If you would like your data points to be more centered, you can add a maximum x and a minimum x value. For example, if I wanted to display all data points between -5 and +5, this will automatically be reflected in my plot. I can adjust the axis name. So on the x-axis, I have the log2 fold change. I can adjust the font size to be larger or smaller. And I can also choose to rotate this 90 degrees or keep it perfectly horizontal. The labels can also be rotated as well. And the same can be done on the y-axis. Once I've fully customized my x and my y-axis to my liking, the next thing that I might want to do is customize the color. So to do that, I can add a visual map. And this can be either on the basis of what you have on the x or on the y-axis. So typically the color changes correspond to the log2 fold change values. Perhaps all of the values that are considered upregulated are painted in one color, and values that are downregulated are painted in another color. So to do something like this, I would select log2 fold change for my dimension. And now I'm going to specify what are the maximum and the minimum log2 fold change values for each color. For example, if I want everything greater than 0 to be blue, and a maximum value of 5, I can leave that here, and now I have to add a second color for everything less than 0. So to do that, I would add a color, specify what the minimum value is, and the maximum's value would be 0, and I can change this to red. So I could add as many or as few colors as I would like, and this would be reflected in my plot. The next thing that I can do is add a title. I can also remove the grid lines if I don't want them to show, and again, I can download the plot at any time and customize the image type as well as the resolution of the download. Once you've taken a look at your entire data set and visualized it, the next thing that you might want to do is filter your data and take a closer look at what genes are upregulated or downregulated. To do this, go to your data table and use the drop-down menu located next to each column header. You have the option of clicking on the column headers and sorting from greatest to least, so you can quickly take a look at what genes have the highest or the lowest log2 fold change. And you can also apply a filter by clicking the drop-down arrow. You can filter by a condition that is greater than or equal to, greater than, less than or equal to, or is between. So if you wanted to look at the genes that were upregulated, you could do greater than or equal to any log2 fold change value that you would deem as appropriate. For example, all genes with a log2 fold change greater than or equal to 3. This will automatically subset my data set, and this will be also reflected in my Volcano plot. And now I can adjust the axes, so now that I can see things that are just upregulated, I might do the minimum is 0, and I can remove the maximum. So now I can see all of the genes that have a log2 fold change greater than or equal to 3. I can also zoom in to take a closer look at these genes as well. Once I've taken a look at my genes, the next thing that I might want to do is look at the insights. So of the genes that I've subsetted, everything with a log2 fold change greater than or equal to 4, which pathways are enriched? To find out this information, you can click on the Insights tab. On the Insights tab, you'll be running a gene set enrichment just on the genes that you filtered and are currently found in your data table. And again, we break these down into several different categories. And you can click through and see exactly which pathways are involved or upregulated in the gene set that you have filtered. For example, if I click on one of these pathways, I'll be able to see all of the genes that are involved in this pathway as well as in the data that I filtered in my data table. I can take a look at the description to get a better understanding of what this gene does, and again look at the observations. So once you've done this, you might see a gene or a pathway that you find very interesting. To learn a little bit more about specific genes or insights, you can head over to our knowledge engine. So here you could search in a specific term or a gene that you're interested in, perhaps one that you saw within your insights. Enter your search term of interest and confirm exactly what you're looking for. So I'm interested in BRCA1, the gene. Once you confirm, you'll be taken to an overview page, and you can head over to the Papers tab, where you'll get a list of all of the papers associated with your search query, and you can sort this on the basis of publication type as well as publication date. Under the Genes tab, you'll get in-depth information of all the genes associated or found in your search. Heading over to Detailed View provides you with an in-depth summary of all the information you'd want to know about a specific gene. Under gene sets, we break down all of the gene sets that this gene in particular is associated with. This is sorted according to categories. And as you click on each category, you get a breakdown of all of the gene sets associated with the category and all of the pathways found in each gene set, with a description of what the pathway is and the role of your gene of interest in this specific pathway. Under observations, you can see any observations you have of that specific gene and all of the data sets you have either generated or uploaded to the platform. Under annotations, we provide you with a list of all the papers that give you insight into the reference into function of your gene of interest, and you can visit the link at any point in time. Under Gene Network, we provide an interactive gene network map that shows you all the interactions with your gene of interest. And we break down the relationships between each one of these genes according to whether it catalyzes, reacts with, activates, inhibits, or binds with your gene of interest. Under disease, we provide you with information in regards to any diseases that are associated with your gene of interest, whether they're causal or correlated. This is further broken down into the specific relationship, whether it contributes to as a pathogenic condition or is causal for, as well as all the different diseases that your gene is associated with. So now you have uploaded the data, organized and processed the data running a bioinformatic pipeline, visualized your data using heat maps as well as Volcano plots, conducted a gene set enrichment and looked at all the pathways that are differentially expressed or upregulated and downregulated in your experimental and reference samples, and also did a little bit of a literature review and looked into public data that's available to learn more about your data. And that is how you do a comprehensive RNA sequencing analysis from start to finish on the BioBox platform.